All right, so we're going to be learning about epithelial tissue today. And so the point of this video is to sort of give you a very quick overview of the different tissue types that you'll be observing in lab. There are 7 different epithelial tissue types that we are learning in lab. There are a few others that we may talk about in lecture. A couple of the defining features about the epithelial, or the ET layers, is they are tightly packed cells. There's very little space between these cells. They're often found lining the inner and outer surface of organs in the body itself. And so the epithelial layer, tightly packed cells, and they have a free surface. What that means is that one side of that tissue layer is in contact with another tissue, but the other side is free. And so again, the fact that these are covering or lining organs means that there should be a free surface. And so if we go through these in order that you find in your lab manual, the first example we have is the simple squamous ET. And the name here, we'll see, is meaningful. The word simple at the beginning is indicating a single layer of cells, whereas the second part, the squamous, is describing the shape of the cells. And that term squamous is describing a very flat or sheet-like cell. And so here, this is a blood vessel. This is the example we have for you in lab. And we know that there's a few things we can see here is blood. And so these are red blood cells. That's kind of a nice feature to tell us that this is in fact a blood vessel. This would then, you can imagine, be the wall of the blood vessel. This is the center where the blood passes. We are looking for the innermost layer of a blood vessel. That is where we find simple squamous ET. There are a lot of other places in the body that you should be familiar with, but the example we have in lab is this, the inner lining of blood vessels. Now, if you'll notice in this slide, here is this very dark purple band. This is not the epithelial layer. This is some elastic tissue that we'll learn about later. We should be looking just inside that. This very thin layer, kind of a lighter pink tissue, this is the epithelial layer. And often, because it's so thin, the thing that becomes something we can see would be the nuclei. So in certain areas, we see this bulge out a bit. And those bulges actually are the nuclei of that cell layer. And so simple squamous is very, very thin. And it's so thin that at times you don't even see it, except for in areas where we have these nuclei. And so the simple squamous, as we'll see when we compare to the other ones, is often difficult to find unless you know exactly where to look because of how inconspicuous it is compared to other tissues. If we move on to the next example, this is a section through kidneys. And we're using the kidney as a really nice example of simple cuboidal. And so the second type of tissue that we'll be learning is the simple cuboidal. And the name is sort of similar to the first one. The simple part means it's a single layer. And the cuboidal is describing the cell type. And so this is a cube-shaped cell, a single layer of cube-shaped cells. And we can see about 7 or so tubes or ducts. And the kidney itself is just made up of millions of these ducts. And in certain stretches, they have these beautiful, beautiful, simple cuboidal ET linings. And so this, I think, is one of the nicest examples of the simple cuboidal. The shape of the cell is absolutely cuboidal. It's not, as we'll see, thin. Now, interestingly, we are not using this slide to study simple squamous like we just saw in the blood vessels. But the truth is a lot of these tubes, a lot of these ducts here are in fact lined with simple squamous, very thin with bulgy nuclei. So these are either tubes of the kidneys or these are small capillaries that are a critical component of the kidney. So you won't be asked to identify the simple squamous layer used in a kidney slide, but just so you know, that's what most of these tubes in this view are. We're using this slide for lab just to highlight these simple cuboidal cell layers. Now, the third one we have here, this is moving down our list. This is going to be an example of a simple columnar ET. And again, the name here is simple, single layer. And so here's this nice single layer. Here's our free surface. And so that is then going to be the epithelial lining. And it's simple, single layer, but the shape of the cell is very nicely columnar. These are tall columns. So here's a cell. Here's another cell, and you'll notice that the nuclei tend to be all lined up in about a single row. And so we have this nice single layer of columnar-shaped cells. Now, the 2 examples that we are using in lab, we have the small intestine, the lining of the small intestine, as well as the stomach. And so this tissue type is very common in that part of the digestive system. Now, there's another feature associated with these particular cells that we find in the small intestine. And if you'll notice along the surface here, there's a kind of a fuzzy border. And that fuzzy border is sometimes called the brush border. And what's creating that is, in fact, really, really, really small cellular extensions referred to as microvilli. And microvilli are sometimes surface features of cells, and they're typically associated with increased surface area for absorption. And given the function of the small intestines, as we'll be learning later, absorption is really critical, and efficient absorption of nutrients is really important. And so it's not surprising that the epithelial lining of the small intestine contains these additional features or microvilli. Now they're really small. We can't see the individual extensions. And so collectively they create this sort of fuzzy brush border, but that's what we're seeing there. Now in the next example, we see another example of simple columnar. This is from the stomach. It may appear a little different. It could be the magnification, but we still see a single layer of cells. In this case, it's sort of following a nice little pit here. And the single layer of cells is composed of columnar-shaped cells. Each one of these cells is shaped like a column. It's not square or cuboidal like we saw previously, and it's definitely not flat or squamous like we saw at the beginning. Again, the nuclei seem to be all arranged in a single layer. Now, that's important because when we look at the fourth example, this is now our fourth example that we're going to be looking at. This is from the trachea. And the layer that we see here is referred to as pseudostratified columnar ET. And the name is interesting because when you look at this, first glance, you notice that the nuclei are not all lined up on the same level. Right? There's some nuclei up here, and then there's a lot of them in this level. There's some down below. And so if you just use the nuclei, which are often really prominent because of how they stain, this appears as though there are multiple layers of cells. And when there are multiple layers of cells, we call this stratified. But the truth is there's only a single layer of cells. It just so happens that the nuclei, because of this sort of inconsistent pattern, appear to exhibit stratified, but in fact they aren't. And so that's why the name pseudostratified is given here. These are falsely stratified. They appear stratified, but the truth is every single one of those cells is touching the basement layer, which is the bottom of the ET, as well as the apical surface, or the lumen in this case, of the trachea. And so we have a single layer, but because it appears stratified, it's referred to as pseudostratified. The columnar part of the name is just describing the cell shape. These are column-shaped cells, very tall and narrow. And then there are two additional features that we see here. You'll notice along the surface of this appears a lot like the microvilli we saw in the previous couple slides. They're in fact not. These are referred to as cilia. And even though cilia and microvilli may appear similar, it turns out that cilia are much larger, they're longer. We can almost start to make out individual filaments or extensions where that was not even possible when we were talking about microvilli. And functionally, they're very important in their differences. Microvilli, as I had mentioned, really important for absorption. Well, it turns out cilia can move. That cilia have the ability to move. And so that movement is associated with moving things, moving stuff, moving contents. And in our respiratory system, where we see this is again the trachea, there are goblet cells. So another unique or additional feature or accessory feature that we see here is the presence of these goblet cells. And these goblet cells secrete mucus. Go back to that, sorry about that. The goblet cells secrete mucus, and they secrete that mucus to the surface. And so you can imagine the surface of the trachea, and really a lot of the transport part of our respiratory system is lined with mucus. And that mucus is important because it traps dust, it traps particles, it traps things that we breathe in. We don't want that following down into our deeper lung tissue. And so the mucus can then be moved, it can be carried up the respiratory tract by the cilia. And so microvilli, which we saw previously in the intestines, is associated with absorption, added surface area. Whereas here we have cilia, and it's the ability to move that then allows these features to move mucus through, which is important in the respiratory system. So we can call this a pseudostratified, ciliated, columnar, epithelial ET with goblet cells. The next two are going to be our true stratified layers, and so everything so far has been a single layer And now we see for the first time a layer of ET that's composed of multiple cells. And so if we look at this is from the esophagus, and this would be the center of the esophagus. This would then serve as the wall of the esophagus. Everything that we see from this point right here out to the surface, this is the epithelial layer. And you'll hopefully be very convinced that this is made up of multiple layers of cells. I mean, here we can see a very discrete cell. And so when we have multiple layers of cells, then we refer to that as a stratified layer. So this is a stratified ET. And when we have stratified ET, then we need to look at the surface cells. The shape of these cells will then give us an idea of the specific type. And these cells near the surface within the esophagus are flat. Right? These are flat, squamous-like cells, and so this ET is referred to as stratified squamous ET. And we see stratified squamous ET here in the esophagus. We see it in our mouth. We'll learn about a slightly different type on the surface of our skin. And so areas where you find stratified ET would be areas where there's a lot more abrasion, a lot more friction, a lot more of the surface cells just regularly sloughing off. And so this serves as protection, right? It's protecting the underlying layers. If we only had a single layer, well, then that surface damage would quickly extend down into the more supportive, and then eventually down to other tissue layers. And so stratified ET is for protection, and here we see a stratified squamous. This is the main one that we're going to learn in lab. The next slide is actually of our sixth ET type that we're learning in lab, and this is also a stratified layer. But it has a unique name. It's called transitional ET, transitional epithelial tissue. And the reason for that is this is the type of ET we see lining our bladder and other aspects of our urinary system. And if you think about the function of the bladder, it's to store urine, and therefore it needs to expand as urine increases, and then sometimes it'll empty and shrink back down. And so things that need to expand need to have a surface layer that can stretch without tearing. And so here is a stratified layer, it goes from here to here. And the truth is that the surface cells will change dramatically depending on if this is stretched out or if it's relaxed. And in this example we see here, it's pretty relaxed. These surface cells are kind of cuboidal shaped. They're not really flat, not nearly as flat as we just saw. And so this ET layer that's stratified, but that can change the surface cell shape based on stretching, is referred to as transitional ET. The final ET layer that we have is a little different. It is the glandular ET. Now I mentioned that one of the defining features of ET is this free surface. These appear to not have a free surface. This just looks like tightly packed, grainy looking cells without any obvious free surface. Well, we'll get to that and we'll talk about more of that in the lecture. But the truth is, these are ET. And hopefully you'll be convinced later on that they do have a free surface. But this is a salivary gland, right? The sweat glands. Glands are modified ET, and the cells that we see here, most of the cells that we see here are in fact the cells that secrete the contents. Maybe it's mucus, maybe it's saliva, maybe it's sweat, whatever it may be. These are the secretory cells. They have to secrete their contents eventually to the surface of the body or to the surface of maybe an organ. And so there are ducts. There are more familiar looking ET layers spread throughout. And this one only has a few up in the top right, maybe down here. And so as you look at these slides, recognize, and I think it's important to note, that there are always different cell types within the same field of view. And that's just the nature of taking samples from an organ. It's composed of all the tissue types typically. And so when you look at something like this, this is glandular ET. This is the secretory stuff. Whereas these represent the ducts of those glands that transport the contents to the surface. And typically, these ducts are going to be composed of simple cuboidal. And so these may look familiar. They may have that simple cuboidal shape, and that's because simple cuboidal is one of the more common tissue types that we find in the ducts of our salivary glands. And so just kind of a last thing to emphasize here is in no way should these 7 examples or 8 examples be the ones that you study. it's really important that you observe the diversity. It's really important that you recognize that tissue types can look very different, and that it's the hardest thing is to identify these tissue types really amongst the sea of other tissue types. And so use this as sort of an opportunity to be familiar with the material before lab, but definitely use the time in lab to really become familiar with the different types of tissues and the variation that we see between them. So hopefully